Hello guys, today we're going to talk about DNA replication. So just to recap on why we needed DNA replicated, as you know, when a cell division occurs, uh, the two new cells need to have the same amount of DNA. So there's a need for the DNA to be replicated. So one strand of DNA, so like right here, so this is double helix strand, and when it get duplicated, the new strands also join the old strands and they get separated. So that's how this is what DNA replication is. So there are a lot of terms that we're going to describe in this lecture, like ORT, origin of replication. ORT is where the replication starts. So on a DNA, there could be many points that origin of application can occur. So I'm just showing two. There could be more than two. So what is a helicase? Next term is a helicase. Helicase is it joins here on both ends of the DNA and try to unwind it. So see how separated it is from this direction to this direction. So this separation is caused by this helicase on the both ends, this end and this end. So that's the role of the helicase. What is a DNA polymerase? It adds nucleotides to the three end of the new strand, but it has to have a RNA primer. So, so this is a DNA polymerase I showed you in here in the picture. This box right here that I made is a DNA polymerase. So it joins at a location where the DNA replication is going to be started. So there are two leading strand. One is leading strand right here, and second one is right here. So this is where DNA polymerase would join and start adding nucleotides from 5 prime to the 3 prime direction. From 5 prime to 3 prime direction. See how opposite they are? Because when this replication completes, this 3 prime end would be complementary to this 5 prime. Okay, and this 3 prime end would be complementary to 5 prime end. Now you are asking, okay, half of the DNA is on the leading strand is done, but what about this strand? How we replicate right here? I'll show you in the next picture. So those are called lagging strands. Lagging strands. So let's move on to the lagging strands. So there's a requirement that DNA polymerase, which is right here, it also joins right over here. It only does the replication from 5 prime to 3 prime end. Okay, and that's the only direction it moves to. So for it to move from 5 prime to 3 prime end, as soon as helicase unwind this part of the DNA, it joins right here. And RNA primer, which is this red section, is RNA primer, this one. RNA primer is already been done by RNA polymerase. So before DNA polymerase joins to this location, RNA polymerase already had this primer made for DNA polymerase to start doing that application. And then it start moving to this direction. Okay, and once it ends right here, And as helicase unwind more DNA in this direction, it jumps off and join right here, and then start doing another replication in this direction. So now you can ask me, and it's the same scenario occurs on the other lagging strand. So this is another lagging strand right here. So these are called, there are little spaces left in between. These are called Okazaki fragments. This is the guy who figured this out. It happens due to because DNA polymerase only moves from 5' to 3' direction. Okay, so that is the reason it jumps off and leaves these fragments, and these fragments are joined later by enzyme called DNA ligase. It joins the fragments and create a continuous strand. So that's the theory behind all this replication. So just to recap, that there are two strands when helicase unwinds the DNA on both the directions. Helicase right here. There are two strands, leading strand and lagging strand. Lagging strand and leading strand. And DNA polymerase attaches at the point of origin, which is this, and moves from 5' to 3' direction. 5' to 3' direction. Same here for the Okazaki fragments, 5' to 3' direction. Now, once this whole scenario completes, you will end up having DNA replicated. Each strand would have an exact copy of its complementary strand. Hopefully you like this lecture, guys. If you like it, go ahead and make a comment and subscribe the channel. We'll see you in next tutorial.